Hi everyone. Welcome to our third session. First and foremost, a lucky find by Mr. Corfield with some amazing material with lots of fungi in it. And Adam did some counts today, and there's like 1,300 micrograms per milligram per milliliter of material of fungi, and a 1-to-1, fungal to bacterial biomass ratio. Perfect, really. We can boost it with protozoa and nematodes, and you're going to have probably some of the better materials, that some of the best materials that we've seen, in the country. And I think the magic ingredient in the production of that material was time, wasn't it, Niels?
Yep, we got some seriously aged wood chip there. Pure, what is it? Basically, it's pure hardwood chip from a forestry yard that it varies from the original scrapings to more modern ones, basically, but it's from a yard that was taken on in the 70s and has never been scraped until I collected what was there. So there's everything from like 10 to 40-year-old material there, basically.
That sounds like cheating to me.
I just leave it there, shall I?
So yeah, but it seems like, things like, fines like that, obviously, these are not something that we would need to set up a system. It's something that my guys here suggested, that we should manage woodland in this sort of way, where we can have this material actually breaking down, in pretty much in woodland, and sort of have like a multi-year cycle where we have this mother culture. And it's something to consider. It's something that we might consider for the future, because there are places which could be managed like that, and I don't see why not. But I think James was on the call. You might be a client for Niels for the trials that we're planning. Perhaps we might also try and grab some of that material from Niels and try that with your trees. I don't know, Niels, if you've ever met James, but he's a top fruit grower in Kent. So we're looking to run some very minimal trials, because this is untested, uncharted territory. So we never want to risk anything in the beginning. So we'll be looking at 5 trees here and there, kind of thing, in terms of the design of the trials. So and then once we go through a whole 1 year cycle and we don't kill any trees, and we show results, then we can look at something wider than that. So that's the opportunity there, in terms of top fruit. But with this amount of fungi, I think it's something that would be worthwhile to talk about, to set that up. So yeah, so that's Niels's numbers, and I'll put them into our leaderboard, and I'll put in the notes cheater as well, just to make sure that everybody is happy with that. But great find, Niels. I really like that.
I think it probably is worth saying that there are other. This isn't that uncommon for forestry yards, actually. To be honest, there's normally like a corner or an area of a yard that hasn't really been touched, just because it's just a waste material. So they do the processing, whether it's logs, milling, whatever. Basically, they push up that material into a corner and just keep doing that, and it just gets sort of further and further into the edge of the yard. So I would just keep an eye out for anyone that is doing forestry work, and just ask to have a look around the yard, basically, because actually often this material just is, if you can get it out, and often I think that's one of the issues, like kind of inaccessible spaces. They might have been sort of tipped down a bank or something like that. You know, getting it out is probably the main issue basically, but in that instance you've got great starter culture, great bedding for worms. So yeah.
No, it's a good call. It does sound like, you know, finding a an island in the Pacific that basically is inhabited only by birds for the past 3,000 years, and you've got the amount of, you know, guano over there is allowing you to mine for phosphorus basically. But then it's going to run out at some point. So, but it's something in the interim. I think it's a great idea to have. And especially as you call it, mother culture, you know, just basically have, because what we're finding obviously is the longer the material keeps breaking down, the bigger the chance that you're actually going to have the microfungi that are the guys that will then feed off the plant exudates, not the guys that feed just on the lignin of, you know, like the guys that, you know, first come in and break down the lignin kind of thing, but the ones that come later afterwards. So yeah, brilliant stuff. I think this is something to look into. I've got I have not finished. I was hoping that I would have this ready for today, but I haven't finished because I wanted to present to you the design of trials for next year. The theme throughout these calls will be always building a consortium for trials for next year. So officially, we're building this this case for a grant, and we'll be going with Sheffield University and with NIAB, and we need to have a consortium of, you know, of about 50 farmers willing to do trials with us. And so what I'm trying to do is to design these trials in a manner that will be a no-brainer for the farmer. And so I've been doing some calculations, and I've been doing some drawing up as well. I still want to go for something that is more than a little plot of land. So every trial would be you know, of decent size, to ensure that we're not just looking at, you know, the interaction with microbes with individual plants, but we're actually looking at what needs to happen for this to be a reality at large scale. So we want to push the boundaries, you know, what equipment is needed, you know, what modifications we need to do, and all these things. And so the trial design basically is that the total biology area is minimum of 5 hectares, and then it has to be matched with the conventional side used on the farm, and then with a control. And my biggest gripe with that design, obviously, is that the control side of thing loses you the most money. And so I've been working on this conundrum, and I've got my notes here. I need to put this into a presentation. Once I've got it ready, I'm going to circulate that to everybody. But the idea is that in terms of output yield, if we're doing this on spring barley, because we're doing this on spring crops, and again, Judith, we're going to have a separate conversation for your design, because I haven't got a design yet. You know, we need to work this out. And obviously with James on trees, that's a completely different design. But in general, if we're looking at spring crops, we're looking at, I was making some assumptions. I was making assumptions that the conventional side for many conventional farmers would be spending £500 a hectare on inputs. And then we have our cost for inputs at £600, and then cost of inputs on the control is 0. And this doesn't take into account harvesting. It doesn't take into account all the other costs. But then I looked at the feed barley prices, which are last were at £188 a ton. And so then I'm making another assumption that the conventional side will be yielding between 5 and 7 tons a hectare. So I need feedback from you guys as well, what my assumptions are. And then on the biology side, yielding between 4 and 5 tons, being conservative, and then 2 tons on the control. And we don't know if that's not going to be a write-off as well. So we need to basically look into that closely. And then that would basically give us a sort of net profit, net calculation, for conventional per hectare of 816 pounds, 340 pounds on the bio, and 376 pounds on the control. And what I'm trying to get answered before, with this grant is how can we use this grant to basically pay the balance to the farmers participating. So the grant, because we're a micro-organization, the grant can cover up to 70% of our costs. And that would basically mean that now with the grant, the biology would be producing about 760 pounds net. And then, so that's the more comparable with the conventional side. And obviously, we're still going to be aiming for matching the yields. That's our whole thing. We want to match the yields. So I'm being very, very, I can't hear Adam, he's pointing his finger, but what I'm basically trying to say is that we want to basically match, and hopefully I think Adam is saying that we want to go above that, but I am being very, very conservative with this whole assumption. I'm trying to make a financial case for the grant with the worst possible outcome. So the one question I still haven't got answered, and I need to speak to KTN, which work with these grants, is can this grant cover the difference in income on the control side? Because if they cover 70% of our costs on the biology, then basically the result is that you're making money on the biology side of the trial. If they then can cover the difference on the control, then you don't lose any money during the whole trial. There's nothing that you basically you're losing at this, and you pretty much have got a lot of you pretty much have got very similar kind of income profile as the rest of the field. So that's basically how I want to design these trials, that we have large enough scale to push us into using large scale machinery and simulate what you would be doing normally, but also in terms of the trial, there's really no risk in terms of money on that field for the farmer. So that's what we want to where we want to get to. And then there's the last question that Judith will be asking: how much do the sampling costs, right? So we'll be involving NIAB, we'll be involving Sheffield. Their costs are fully covered, 100%. So we don't worry about them at all. There's then basically, our costs of our microscopy. And there's also one more question that we need to answer. Do we need to use another provider for microscopy because we're using our product? So I need that question answered. There is another lab, that's Eddie Bailey, who does analysis, which is the same as we do. So that's another question. But in terms of cost, I need another question answered by Sheffield University. Do we need to do samples on all 3 replicates of the trial, or can we do it on one replicate? And that basically creates, a huge difference in terms of cost. Because I believe that we would need to have at least 9 samples across one replicate. 5 samples on the biology, 2 samples on the control, and 2 samples on the conventional, 2 samples on the control. That's a minimum of 9. So I rounded up to 10. And if we have to do it on 3, then that cost is £1,650, reduced by 70% of the grant. That's now £495. But if we can do it just on one replicate, the cost is £165. So that shouldn't be a problem for most people, basically across the growing season, because it's minimal. I hope so. So in general, basically, if we can play this right, the cost to the farmer to run this trial will be 165 quid, pretty much. So that's basically, in general, the outlay throughout the season, and then sort of like, how this all works out basically. But in general, it should all work out to about 165 quid. But I need to go through these numbers again, make the presentation, make the case. And then once I've got the case, then I can ask you to go far and wide and, make the decision, who's going into this consortium, because it's this consortium that will be able to convince the grant providers to give us the money. And so that's it in terms of my update for today. We're basically pushing forward for that grant. The deadline is the 1st of December. So hopefully towards the end of this week, I'll have everything ready, pushed out to everybody, and I will be very happy to get your feedback and questions on that. And does it even make sense? Over to you.
Now, if nobody else is going to say anything, it makes complete sense to me, but I think as a scientist, I would want the triple replicates of everything, at least at the start, in an early test. I don't think you'll get away with only doing a third of them.
This is definitely something that we need to get it cleared properly, so yeah. I'll get hopefully that cleared, and I'm just trying to make sure that
I understand, but it would be a huge disaster if we did it and it all worked and then they said, well, it's not good enough, you haven't done enough testing. That would be the worst of all worlds. We
need to have it properly signed off by the University Boffins, and once they're happy, then we'll have to go with that, where they're going to basically prescribe to us.
Mm-hmm.
Yeah, what are they saying on that front in terms of replications and statistical significance?
Well, the first requirement is to make sure that we've got everything in threes. So we need to have conventional biology control, same size, repeated 3 times.
On each farm?
On each farm.
Yeah, right, okay. Mm-hmm.
Yeah. So then we have, and this is basically, then every trial can be, treated separately, and that's basically, because we're pretty much testing the biology against individual farm system. Everybody's got a different system.
Daniel,
Yes.
When you did the testing, you talking about microbial biomass? What testing are you talking about? The soil test.
For us always it's the it's the testing that we do with the microscope assays that we do for bacteria, fungi, protozoa, nematodes. This is the regular test that we use. In terms of other testing that we want to include, we want, and I don't know how much of it we can do, but we want DNA analysis of what's in the soil, what we're putting out, what's surviving. And I don't know, again, what sort of amount of work that's going to require. So I need to clear that with Sheffield. So hopefully I'm going to have that answered by the end of the week as well.
So you want to know the DNA analysis?
Yeah.
And how many tests does that amount to from the potential trials you're looking at?
I need to basically speak to Sheffield about the numbers, and so you know, this will be the bulk of their work, just sitting on the PCR machine, just to make sure that, because I really want to know, you know, we've got one of the things that we simply don't know is what do we produce, because it's just something that we look at morphology of. And so it will be interesting to understand, you know, what we produce actually is surviving in the soil and actually contributing. So there's a lot of papers already that are species specific. And so if we can then cross-examine, you know, what is actually happening during these trials with these papers, then we can have a massive body of evidence for, you know, for how soil biology, you know, interacts very, very properly, because we're going to be collecting data from real life, you know, situations here. This is why this large scale stuff is important to me, that we can have this real, you know, burn test here.
And that before and after, presumably.
Say again, please.
That would be before and after.
Yes, exactly. You know, throughout.
Yeah.
This is something that we need to design with Sheffield, and you know, they need to be the driver of that.
Yeah, good stuff.
Awesome.
So are you looking to like assert what biology is there before, and then, I don't know, 2 or 3 weeks after you've applied it, what's like established, and then what's at the end of the season or mid-season at some point later in the year?
Yeah, that will be basically useful. You know, it's again, I don't know how much we can do in terms of the DNA analysis. I'd like to do as much as we can, but we, you know, with our microscopy, we have to take samples 2 weeks after each application, basically, because then we have an answer of what survived, and
right.
But then we just base it on the morphology on the groups. So.
Mm-hmm.
But yeah, we want to understand, you know, what is actually happening. The DNA analysis is also necessary for the bacterial profile. You know, there's ways to work with bacteria that doesn't require DNA analysis, and probably, you know, University of Sheffield or other labs have got the gear to grow out bacteria that are found in these things, but it's just it's not as efficient, in my opinion. You know, from what I understand, you would need to have, you know, separate formulations of, you know, petri dishes to grow the colonies each time, and you spend, you know, a lot of time to grow them out, and then you'll be missing species. So I think DNA analysis in this case would be the best, but again, that's something that I would get Sheffield to make a decision on. We just want to know what's there. We just want to understand what is actually doing the work. Do we have, you know, all of the nitrogen fixers? Do we have the phosphorus solubilizers? You know, do we have the AMF fungi? And, you know, we'll be investing also in the equipment that will allow us to do colonization analysis for roots. So all these things are important to us, and we just want to collect all of that data. It's going to be a massive, massive data collection exercise for the whole thing. But it's, you know, the whole point is, if, you know, ELMS and the government and the drive towards, you know, soil health, you know, if we are to answer those questions, you know, what is actually doing the work, we need to do this work, and we need to do it in the field. I think, you know, there's been a lot of studies, but once we do it in the field and we actually say, okay, in the field, this is actually what's happening, and we have an answer to that, I think then we have a basis for a complete shift in agriculture. And we can then really present this to anybody. And we can say the UK is a pioneer in basically regenerative agriculture. And, you know, you want to be regenerative, be regenerative. You want to go organic, go organic. You know, want to stay as you are, stay as you are. But it's then you've got full picture. There's no guessing anymore. There's no dogma. We want to get rid of the dogma, basically.
Is there any trials along these lines that have been done elsewhere in Europe, North America, Australia, wherever?
Probably not at this scale.
So they've just done it on little trial plots rather than field scale.
I've been trying to find papers that actually, you know, would have that, and I found one, for instance, in the Netherlands, and it was very inconclusive because of the two problems that we're trying to solve is, one, access to this biology, you know, and reliability of the inoculum, and the second one is then the trials. And the first hurdle is where people stop. And then basically they just stop at this, you know, reliability of the inoculum, and they don't really know what's in it. And so if we're applying something, we need to basically have a full DNA analysis, full scan, then we know, you know, we go by species. And then we have, you know, a full profile. And then we can say, okay, well, if that field was inoculated with this batch, but that field was inoculated with that batch, and we've got a full profile of everything that we've put in it, then we've got an answer of, you know, across 50 farms and across different soil types, we have a much better idea of what is going to want to work on which soil profile and which crop. So that's why, you know, it's not enough, you know, in my view, it's just not enough to go by anecdotes by other farmers across the world. You know, there's been so many fantastic early adopters who took and run and did as much as they could with compost, you know, with compost teas and extracts, but most of them are just flying blind, and you know, they've done enough for themselves, but it's not enough for others to be able to simply take and adopt. So we need to go through this whole exercise to be able to answer those questions. Now, you say you, as a scientist, Judith, did you say you've got a scientific background? Did you work in academia? Or?
I did a degree in agric too long ago to count, 1980s. That's as far as my science goes, but I kind of understand what it's about. I suppose I do have a scientific mind, but yeah, I understand the basics of trials and having to have enough and to make sense of it. But to me, the reason this is important at this point in time is that farming actually holds a lot of the answer to climate change problems, and regenerative agriculture is that answer, or could be that answer. And we need to prove it, because I don't see the developed and developing world racking back their emissions enough to make a difference quick enough, but racking back their emissions as fast as they can, along with agriculture pumping as much carbon back into the soil where it should be. The combination of the two could make that difference and could swing the balance. And we need to find ways of encouraging as many farmers to do it as quickly as possible. And if this trial and trials like this are going to allow us to prove that it works, A, that it works, and B, how to do it, and you know, that we can actually put it onto a field scale, then for me it's important that we do it, because no farmer can turn their whole farm over in one go. We can turn a bit of a field over the first year and get our heads around it, and then maybe after 2 years double it, and maybe after 2 years double it. But that's the early adopters, and that's only us here, and a very small percentage of the whole farming population. So the faster we can prove it and get others to join, then, you know, we're doing our bit to try and undo damage that's been done over the last 2,000 years.
Yeah, this is, you know, I wonder if Richard and Doug want to comment on some of that.
What are you getting at, Daniel? About how we encourage more people to do it?
Yeah, why not?
Yeah, well, I mean, what we're doing here is, and I was just about to copy and paste something onto the chat, if I can work out how to get an attachment on there. about the work we've done so far with Kent University, looking at the role of soil microbiome and it within a regenerative agriculture context. And that's all feeding into our work where we are establishing projects to that basically sequester CO2, and those projects can be anything from a whole-scale adoption of regenerative agriculture through to tree planting, wetland creation, whatever. Basically any nature-based intervention that we can measure, and how we reward farmers for that. And basically what we've done is we've established a system that's up for testing now, which is doing exactly that, paying farmers for turning to regenerative practices. It's early days, but it's quite exciting stuff.
Brilliant.
Watch this space is all I'll add.
It's pretty much where we are at the moment. So, you know, building all these things out.
Yeah.
Stephen, you placed something in the chat. What did you find?
Yes, it was something that was sent through to me this morning from RM, and I just thought it was interesting. It was, I think, fairly obvious actually to most of us, but interesting that they have observed, looking at 1,500 different farm samples, that carbon levels are greater where they don't disturb the soil. But,
well, that's the thing, and this is, you know, this is where regenerative agronomists will have to shine there, because you have to have a full system. We can come in with the biology, but if the biology is not supported at some point in time, then it's not going to function when the time comes and it's actually required. So, you know, having cover crops, you know, having fully work. This is actually something that I was having a conversation with a colleague of mine, and he suggested, this is before my time in agriculture, but I'm sure somebody can comment here. Is it true that in the 90s, oilseed rape was 2 meters high?
Yeah.
Because I think Adam had an interesting thought about that, actually. If I can get him to turn around, and if you unmute him.
I was just about to tuck into a new sample. You're stopping me from working.
I'm sorry. I know you've been doing crossover day today already.
This is work.
Yeah, if we can cultivate, you know, varieties that are taller, go back to that old seed stock and try and find those varieties, then you can install a cover crop, a permanent cover crop below them, and harvest just the top. Was the idea.
So yeah, it's a bit of a left field kind of thing, but it came to me because I saw that a combine harvester can be set, you know, the header can be set quite high to harvest hemp seeds, and nobody wants to have hemp growing, you know, any shorter, because you still want the stalk, you still want the fiber, right? So if you can do it with existing machinery, then what would happen if, instead of trying to find cover crops which are growing low, actually have the actual crop growing higher, creating more biomass, and just basically have, you know, the understory just growing to normal height. What do you think of that?
No, that the living mulch sort of idea. I mean, the innovative farmers group have just done a big test trial on it, which is worth looking into, which they've found to be quite successful in terms of overall margins from the crop, and then having the grazing. They're using a micro. God, I've forgotten the name. Micro something under the grain crop, and overall, they're getting better farm profitability because they're combining with the grazing, but they're having some issues with perennial weeds, but none with annual weeds. So it's, you know, it is an area I think a lot of people are looking at. And then you've got the work of Farm Wild or Wild Farmed, whatever, you know, I don't know if anyone's heard of Andy Cato and what he's been doing, but he's doing, you know, pasture raised crops, basically growing crops through pasture, and he did it very successfully in France after years of trial and error. So there's people giving it a good go, and I know I caught John Pawsey talking at Groundswell, and he was talking about a new machine. He's just got a new drill. He's just got commissioned from Sweden that is going to do this in literally within sort of millimeters new seeding for him, where he can move the living mulch mat basically throughout the rotations to alternate what's growing in between. It will require a minimal amount of tillage, a very, very low tillage, but not every year, and just in between the crops. So there are some really interesting trials going on the moment. But I was just wondering also, Daniel, if you've said this grant is obviously from the UK, but I mean, is the trials that you're looking to set up looking internationally as well, just thinking about covering different soil types, proving this in the greatest number of scenarios, and also you know, you get second seasons, you get to try twice in 1 year, or, you know, more times depending on where you have sister sites internationally.
You know what, Harry, this is very ambitious what you just said, you know, because I think it's something that I'm sure we'll be interested in, but probably not next year. You know, we're just a small team here, and we just need to understand, you know, where our feet are and how we can walk, and then we can see if we can speed up that walk a bit. So this whole trial for next year, what we're trying to pull off, it's still a big undertaking for us. If we couldn't have, you know, people like NIAB and Sheffield on board, we wouldn't be able to manage it even. So for us the you know, the important bit is that we can do all this stuff on a fairly, you know, on a national scale, and you know, with a decent number of farmers, basically. That's the first thing, and then we can talk about going elsewhere.
Yeah, well, just sometimes these universities have sister universities in different countries, and, you know, they might be able to, Sheffield might be able to open a door with you with their sister university in, you know, the States or Australia or something. And
that's very true. The limiting factor here is access to biology, and so if we're producing the biology here, obviously we can't ship it, so we would need to produce it elsewhere, and that's going to take time. So it's not something we could do next year.
Thank you.
We've got probably about another 15 minutes, and these sessions are also, you know, should be providing some value for people who are making compost. Are there any questions in terms of growing out this stuff that you might have? Because, you know, we're here, we can answer those questions. We don't want to, you know, put a big barrier in front and say, you know, just any questions you've got about growing out your own stuff, go for it.
One question I had was about umai seeds, and just I guess target levels in reports. Yeah, what are we looking for with them, and what do the different levels generally indicate?
Adam, do you want to take that?
Can do. Yeah, it's probably indicating waterlogged soils.
If they're high, sorry, but which values? High values or low values?
Levels being higher, they're indicative of waterlogged soils.
So whereas low values are better.
Sorry, say that again.
So low values are best. That's what we're saying in this case. The target value is 0.
Yeah, they're linked with pathogenic stuff, not necessarily always pathogenic, but yeah, they're early successional decomposers, often in waterlogged areas. You'd want some more beneficial fungi, yeah, for sure. And they've just been reclassified as protozoa as well, so they're interesting things. They look filamentous, but they don't necessarily have the same kind of metabolic processes as the beneficial soil fungi we're looking for.
So they're anaerobes in this case, basically.
Or facultative, yeah. Maybe not anaerobic truly, but on the line,
Obligate.
as the environment becomes flooded, obviously, usually there's less oxygen in the soil. If you think as succession moves towards the forest, the soil becomes a lot more aggregated, both micro and macro aggregates, and it allows for deeper penetration of air. Often the canopy stops water, like, you know, waterlogging happen. If you go for a forest, you'll hardly ever see waterlogging apart from on the paths where humans have compacted, right? So you think the opposite to that is a kind of an alkaline, waterlogged environment. That's where the umycetes will often pop up, but they can sometimes be in the mulch layers of forests, and I think sometimes then they're a bit different. They may appear the same, but they're not quite the same. They just have similar morphological characteristics. There's a lot to be learned about fungi and filamentous organisms. You know, we try our best, but it's not much information out there. But powdery mildew is, you know, linked with omycetes, stuff like this. So, and powdery mildew definitely happens in waterlogged environments.
And there's the case also of, you know, having competition. So if you have other fungi, it's usually also a different environment. So that's why we're hoping that, and I say hoping because we need to run these trials, that if we inject, you know, more fungi that we grow out or find, like in your case, we'll be able to shift these conditions quicker.
Yeah, and if you think the beneficial fungi, they sequester more energy into structure, their ability to sequester water with that structure is dramatically different to omycetes, who will go to spore a lot quicker. Their life cycle is a lot faster. They're opportunistic. So yeah, absolutely, we're going to sequester more water, but we're going to create structure at the same time to get aeration in there.
And hopefully that fungi will also help with with that carbon sequestration. So we want to measure that stuff too. When, you know, we will be doing just like Stephen, you know, you've shared this NRM note. All of the trials will be this is something I haven't. I need to. This is something I need to share with as well. Do they do their own, you know, chemical physical analysis as well, or will we need to have, you know, NRM or somebody else to do that? So I need to look into that too. I must have missed it.
Daniel?
Yes, Rob.
I know you teased me about my fascination with horticulture as opposed to arable, although I am fascinated with arable as well. And this is an arable group, it seems, or certainly that way, or certainly field scale cropping. But interestingly, this year, the tomato industry in substrate and glass had a huge problem with powdery mildew.
Right.
And that there may be something in there that's related to this as well. And I also know that there is potentially some work going into substrate in strawberries around using biologicals. Tiptree, for instance, have been doing it for 3 years very successfully. And it may be connected to your powdery mildew conversation about water logging. It may be because in substrate you water to field capacity essentially 100% and then run off normally. And then that runoff is set at a level that is purely to keep the growing medium free of, well, trying to get these electroconductivity level at the right amount. But actually there's talk now and potentially some research going to go into using biologicals to reduce that runoff, potentially reducing taking that grow bag or growing medium to field capacity of water, which may maybe have some positive impact around powdery mildew, if there's a connection there, which I never realized. I always thought actually powdery mildew was a dry atmosphere fungal. It tends to happen in dry, windy conditions, or, humid, not humid, a dry environment, particularly with air movement. And that's it's be interested to see what we can learn from either side of the agricultural path here.
Would you have suggestions for trials with biology? Because I can think of something.
For substrate?
Substrate and also overhead sprays.
Yeah, definitely. Definitely. The tomato industry was in huge trouble this year in the UK. Massive need to rethink, move to biological. And this is where I guess all of these different opportunities come together around. And I think that what we keep highlighting on these calls is that the more we know, the less we know. and it's a very honest way of saying this is great, but work in progress. And actually learning perhaps from the micro situation of the substrate, we got 15 litres of root volume per linear metre, which is nothing. To what can we do and learn about it, and how can we help it? But what Tiptree have done is ahead of many others, because most substrate is all about almost like a life support system, where you just pump in exactly what you think the plant needs by way of NP and K and micronutrients.
The trouble there might be the space of the pots, possibly.
Well, as you
tried to get the microbes to take to perlite, coconut fiber, stuff like this, but it doesn't work as fast as green waste compost or things with higher bacterial, more bacterial diversity. So it's quite challenging. I think you're looking at like 75 litre pots for a tomato to start to keep it happy, but still need lots more information on that stuff. What kind of growing mediums are you talking about, like perlite, vermiculite,
it tends
coconut
to be pure coir, 100% coir.
Just 100%, right?
Hmm.
Okay, yeah, it might be a problem. It might just be a case of bacterial diversity and the size of the pot, because you need a certain amount of nutrient cyclers to provide a heavy cropping plant like a tomato or something. If you have a smaller root space, then you like you say, you need to keep them on the drip of nutrients to supply what they need to grow. So, and
also, did you say that they're basically watered to field capacity in that small space?
Yes, but that's the Essex scientists are suggesting, or I suggested to them as a farm sort of layman, no scientific justification at all. But if you reduce the NP and K to a percentage, at least half, if not more, and replace that with a biological treatment, which could be a mixture of microbes and stimulant, could we reduce the water over watering essentially? And the view was that actually that could be possible. But at your point about volume, is there enough root volume? That would be quite.
I think a hybrid system would still work well, yeah, and would greatly reduce the inputs for sure, and would.
But it would need to. You don't think it could be done in 15 litres per linear metre, 4 plants or whatever it is to.
Yeah, I mean, we still need more information, but with what I've tried for a long time, it's really hard.
Are you familiar with BioLift, Daniel?
Say again, please.
BioLift. Have you heard of BioLift?
No.
So Tiptree using BioLift as a brand. I don't know anything about them. They don't say what's in it. It's a brand of biologicals. That's what they've been using at Tiptree and strawberry successfully. And his view is you can see the difference. So there's something here that could be relevant. And again, it's all I don't know, but it's about learning, isn't it? And how do they perform? What do they do? Maybe we could. I often think that arable farming could learn a hell of a lot from horticulture around precision nutritional management.
No, sounds good. We'll be very interested, to see if there's anything, that we could do in horticulture. Obviously,
it would be definitely keen to get you involved in that, as we talked about in the past. Okay.
Yeah.
But the Belgian mildew is what got me interested in, because I think what we can learn from that fungal is whether it's in the woodland or in a glass house. I think the more we know about it, the better.
Yeah, there's still, as I said, there's still a lot of unknowns. But yeah, if you want to have a conversation about what we could sort of play around with in horticulture, I'm quite open to start that conversation. As you sort of pointed out, yes, it's sort of my ambition to go towards the spring barley, spring oats crops, but because this mainly is logical in the sense that our validation trial was in spring barley. And so we've got a baseline that we can work off. So, but if you look at this picture here in front of you, you've got Judith, who's a pasture farmer, having sheep in Scotland, and then you've got James, who's a apple grower in Kent, and we'll be doing trials there. So we're not close to doing other things. It's just that logically we're going towards these cereals.
Thank you.
Super.
Judith, could I ask Judith a question? Sorry.
Yes.
Mm-hmm.
In a nutshell, Judith, you've been using biologicals for a while, have you? And what would you say is the difference?
No, I haven't.
You haven't, no. Sorry.
No, I've got a trial field in front of the house that I've acquired from my husband and son. I have actually been putting a bit of biology on that, which I don't know if I told Daniel about, but that's using a Jadam system. And my husband calls it Judith's witchcraft. So I've put some on 3 plots on the farm in different parts of the farm. And I really just now have to wait and see what happens next spring. But on those plots, I will be, I've got the bit where there's been biology put on. I've got another bit where I've got nothing on it. And I'll also then have another bit next to it, which has conventional fertilizer added. We don't use a lot of sprays in the grazing pasture, so that doesn't really count for us in the same way. So I'll be doing something similar. I haven't decided if I can afford to do all the testing that Daniel's talking about doing, and I'm saying should be done. But I'll know more next year as to whether. But I haven't been able to do the testing to even know if the biology that I brewed survived to get onto the pasture.
Yeah.
So,
although I hear witches are going to get pardoned in Scotland, so that you should be okay. I hear witches are going to get pardoned in Scotland, so I think you're going to be okay. What would you say is if you obviously caught maybe out still, but the benefit? What do you think you will achieve from a grass?
What I want to achieve is to find a way of doing what we do on the farm with less chemical inputs and less cost, and hopefully be able to maintain something near the same level of production of beef and sheep.
Right. But you're not seeing
we've gone to rotational from conventional, so we are doing rotational grazing on some of the fields. Right. That's been very successful, and I was saying to Daniel earlier tonight that actually we need to reduce our spring application of fertilizer because we can't now cope with the same level of fertilizer and the rotation because it just goes so mad. We can't keep up with, and our fields are steep, so we can't cut. We can't shut paddocks off and then cut them and round bale them. It's too steep to do that. So that's a success. But to me, the next step is to wean ourselves off chemical fertilizers to some extent if we can, and then to go fully regenerative. It would actually be to be grazing more mature swards where the animals eat a much smaller percentage, and there's a lot of the sward trampled into the soil to then feed that soil biology again. So, but it's a case, you can't do it all in one go, at least I can, because you're risking your income. You can't, you know, and if you're risking too much of that in one go, everything's so variable as it is anyway. So it's baby steps.
Sounds like you're doing a great job. Well done.
Can I ask what Jadam inputs you're using, Judith? Are you using JMS or Jadam microbial solution?
Yeah, using JMS, and the microbes are sourced from, well, the ones I've applied are sourced from a woodland on farm that there's been no farm animals grazing and obviously no other inputs. So that's where I got the source material from. I've actually now sourced better stuff, or what I think is possibly better stuff, from my home farm, which is 30 miles away. And you were talking about woodland soils. I mean, I was under a huge beech tree the other day, sorting stuff and walking on it, and it's like a sponge.
Yeah, they always seem to stand out in the woodland around here too, the beech trees.
Stunning, absolutely. Beech and oak, and it's an old estate. You know, not actually from my brother's fields, but from woodland nearby. And I just went to where the oldest oak and beech trees were, and yeah, covered in leaves now. And then you walk on it, it's like a sponge.
The beech nut as well is very oily. We've seen them being just bursting out with fungi in the natural environments around here. It seems like the beech oil.
I got quite a few acorns, but I didn't find any beech nuts. They'd all been eaten by red and grey squirrels, because there are both there at the moment.
Yeah.
But yeah.
Yeah, it'd be great to see what's going on in your JMS, but it's a time-sensitive process, right? So it's a hard one to kind of send us to sample. It's probably not going to arrive the same as it leaves.
No, exactly. Yeah. So I put it on in three plots, one on my mixed herbs, one on an old pasture, and one on the hill pasture. And I'll see what happens next spring. And it'll just be a case of analysing soil on the different plots that have either had GMS on them or are close by but have had nothing.
Yeah. It's a bit anecdotal, but we've just started a compost pile, and we use something that's very similar to JLF, which is Jadam liquid fertilizer, which is basically like your granddad's bucket of comfrey that he just filled with. Yeah,
I've got one of those with all my vegetables from the garden and the mixed herbs, and then another one with our rape crop. Well, it's Swift crop, which is rape cross kale. So I've got 2 buckets of that going as well.
Nice. So then we followed the JLF with our microbes. And the whole pile went to 65 degrees from, like top to bottom. There was no core. It was just the whole pile was at temperature. So it seems like maybe those liquid, digestates followed by microbes could be really good.
Yeah.
It seems to be this interplay between an anaerobic and an aerobic system, whereby you can take, you take the microbes out of the anaerobic system. They die now because they're in an aerobic system. They become this big bloom of food for the aerobic organisms, which we obviously brew up in an actively aerated environment.
When I'm doing my witchcraft with the GMS, making the Jadam microbial solution, I mean, the soil food web thing says you need to bubble oxygen through it. The Jadam one obviously doesn't. Does the Jadam one anaerobic, or is it a slower process that just takes 2 or 3 days to?
I'm very interested in this because it depends. It depends where you draw that water from. If that water came from rainfall that's just came from yesterday, then the dissolved oxygen in it can be a lot higher than if you drew it from a pond or something like that, right?
Yeah.
So where the water comes from, that sets the scene for what's going to happen within 48 hours. If it starts out below 6 parts per million or 4 parts per million, it's going to be an anaerobic process. But if it's fresh rainwater, then maybe that rainwater could hold the dissolved oxygen for a bit longer. then it can be a different process. So there's really no accountability to these natural farming processes yet.
Yeah.
But I think they're really interesting because they seem to work in a lot of cases.
Yeah, I have mine, when I fill the barrel, I've got two 40-gallon drums sort of thing, and I have mine such that it's pouring in and, to me, mixing in oxygen at the same time.
Yeah, that pouring motion does, the swirling motion does, yeah. But for how long, you know, we don't really know yet.
As much oxygen into it as I possibly can. And the other thing is, up here, well, in the heat of summer, we were maybe almost nearly warm enough, but I actually had to heat the water because it was doing nothing.
Yeah. And the trouble is the microbes use the oxygen up very fast.
Yeah.
That's why the why the
Mm.
compost tea process is how it is. But we're not saying it's like completely useless at all, because we've had stinky input sent to us to look at. Smells terrible. My nose completely writes it off. But then I look under the microscope and there is some beneficial factor to that. Maybe not everything in there is beneficial because of the bad smell, but if you take that then and then brew that, you can remove the bad smell and keep the beneficial parts. So that's what we've found with collecting from the wild. Maybe you get something you don't want, but you can, and there's something you do want, and you can keep that by applying selective pressure to the culture.
Yeah.
And that selective pressure in that case is the air.
I'm going to say a couple of things. First of all, thank you guys for making this interesting when we're supposed to be finishing this conversation again. Next week, I'm literally going to basically go like, right, Adam, Judith, start talking, and you just, you know, just start actually going on the technical interesting bits, you know, so that we don't run out of time, because people are so going. Half of us already left because they've got, you know, other things to attend to. So good thing that we're recording this. And the second thing, I'm actually going to use this as a message to the organics recycling group, which is the association, you know, that looks at green waste composting and also anaerobic digestion. Because at the moment in the UK, not in Europe, but in the UK, those two things are completely separate, composting and anaerobic digestion. And our observations, you know, about how the anaerobic process could be a precursor to the aerobic process is something that should be looked into, because it seems that, you know, there's this switch, you know, when anaerobes get wiped off with the oxygen, the nutrients that they've actually accumulated then really help to drive the process of the anaerobic decomposition and building of our soil food web, just like Adam described. And so if the AD industry, if the AD industry was to take notice, you know, this could be quite a valuable thing to look into, in my opinion. But just on this note, I'd like to thank everybody for coming. Again, we ran over time. I'm serious. Next time we're starting with this conversation, and hopefully we can get some more interesting questions following that way. And at the end, I'll be saying, right, let's build this consortium. Super. Thanks everybody for coming in today.
Thank you all.
Thank you, Daniel. Thank you, Adam and team. Take care.
Thank you. Bye.
Bye-bye.